that we talked about last class. Then I'll start talking about specialized sensory receptors. Okay? In the skin, we'll go through those fairly quickly, because those aren't all that important for clinical perhaps, but then we're going to slow down and spend a lot of time talking about the two most important proprioceptors for therapy, of course, which is the Golgi tendon organ and the muscle spindle. So a lot of time talking about the muscle spindle, it's the key receptor in many kind of treatment approaches, looking at spasticity, looking at stretching, looking at strengthening, all this kind of stuff. So we'll spend a lot more time talking about that than the other receptors. Proprioception is not done very well within the textbook. So on your first class folder, there was like 2 or 3 PDF files about proprioception. Very easy to read. Very simple writing, simple. Read those if I confuse you today, which I undoubtedly will. I'll try my best anyway. What we'll do is we'll go for half an hour, 40 minutes, and then we'll stop and go over the exam. I find if we go over the exam at the beginning, people are still flicking through the exam while I'm talking about someone else. So we'll go over the exam at the end for 45 minutes, whatever. I want to go through it slowly, figure out where you went wrong.
Will we be able to keep the exam?
No, I get them back. The idea is for you can come and look at them anytime you want. Okay. You pre-study, you study before the exam. Next exam, you can come and look at office and we can go through those exams anytime you want. But what we do when we go over the exam, you try and find out where I've misgraded you. Have I missed something? Have I, because there's a lot of people, I might have made a mistake here and there. And what we'll do is on the front page, you write down, please regrade question 4, you said this in lecture, you said this when we were going over the exam, but look, I put this, you misgraded me, give me an extra point. And 99% of the time, you will improve your grade. If you think I've misgraded you somewhere, feel free to write it down. I will look at them all again and try to get extra points out of it. The mean was about, I think, for everybody went about 75%, which was basically the same as last year. Very similar. Okay. But it's people getting some poor grades, people getting 95s, 96%. It's a massive range of scores. There's no reason everybody can't be getting 80s and 90s, seriously. It's not rocket science this, it just takes some dedication, some studying. Jennifer's not here, so I'll pick on her. Jennifer, the tutor from OT, most of you probably know her, she did terrible on the first exam. She'd freely admit it, I'm sure. She's right down near the bottom almost. Next exam she got 25% higher. And she carried on getting that 25% higher every exam from then on. She just figured out what she needed to do to pass this horrible class. Okay? This is a hard class, this is in any allied health, you go to nursing, pre-med, whatever, OT, PT, medicine, the hardest class in medical school is your neuroscience class. Everyone will say that. Because there's lots of these hard, weird words. So, this is a master's and a doctoral program. It is not undergrad Mickey Mouse Biology 101 anymore, right? You've got to buck your ideas up and study the heck out of this class if you're doing poorly. But this exam, remember, is a warm-up exam sort of thing. It's not, it's 20 or 14%. And a lot of 80, 90, 85% of your grade is still a get. But if you go into it next exam like the same way as you did this exam, however you did that, and get another 65, 70%, you're going to do poorly. But there's no reason, I say, people who got 60% can't get 85% the next exam. Okay? If you think I'm going too quick, put your hand up. If there's anything that confuses you, put your hand up and I'll over and over and over and over again until you get it. That's the whole point, right? So what we'll do is then we'll finish off talking about, review this stuff, start talking about our receptors and our pathways. See, this basically ends the review. Now we'll sort of go into more detail of the areas we've touched on and elaborate on various things. Okay, so we finished off last class talking about all these horrible words. These are sort of global issues that can happen with a lesion to the cerebral cortex in various places. Apraxia, which you can't see. Of course, that is the inability to perform a skilled action that you've learned. You can't put skills together. You can't coordinate a skill. You can't walk, you can't kick a soccer ball, you can't do whatever. You're not paralyzed, the muscles are okay. It's just a global issue in the cerebral cortex. You can't put a movement together. You can't put the pieces together to produce a movement. Apraxia. Agraphia is kind of a type of that. You can't basically write. You can't put your handwriting together. Aphasias, a whole series of these aphasias, and I'm sure OTs particularly will go over in much more depth later on. In Sonia's class, you go over aphasias a bit more in your neuromuscular course when we look at stroke. But basically it's a problem with the production or reception of language. The symbols associated with language somewhat. So you can either have a sensory or receptor aphasia, a motor or verbal aphasia. Obviously a sensory receptor or sensory receptor aphasia is basically when you can't see or read words, should I say. It would be visual aphasia, obviously. I just look at something on a paper, it looks like a foreign language, even though 2 days earlier before my stroke, I knew exactly what this was. I can read, but now I can't. That'd be a visual aphasia. Or what's another term for that in terms of word blindness? What would that be? Word blindness.
Alexia.
Alexia, without words. Alexia is another term you see thrown in here. Audioaphasia, you can't understand the spoken word. You can't understand language when it's spoken to you. Expressive, Broca's area problem, you can't speak words. Okay, you can't produce language. It's not dysarthria. Dysarthria is where I can't formulate words because of some mechanical problem in the mouth, for example. Broca's aphasia is verbal aphasia. I can't get the words formed in my brain to come out. And this whole series, you can break these aphasias down to various parts of language people will lose, and I'm not going to go over those here, but later on in other classes you will. Things like semantic aphasia, syntactic aphasia, nominal aphasia, etc., etc. A whole series of more subcategories of these aphasias. Agnosia without knowledge. I don't know what I'm looking at. The visual aphasia. Is that a soccer ball, or is it an apple, or is it a chair? I just have no idea what that thing is. People do bizarre things when they've got a visual agnosia. They try to, like open a car door with a comb, or something silly like that. They'll brush their hair with an apple. Weird things. I have no idea what this thing is in my hand. So I don't know what to do with it. And that's the famous book by Oliver Sacks, The Man Who Mistook His Wife for a Hat. Visual agnosia, you don't know what he's looking at. Tactile agnosia, obviously you don't know what you're feeling in your hand. You pick something up without looking at it, I don't know what it is. A severe form of this is neglect. And there's various terms you'll see for that. Hemispatial neglect, hemiagnosia, unilateral neglect, whatever. That's a very, very severe form of agnosia. And this is usually associated with damage to the right angular gyrus, the right hemisphere. And so you get left side neglect most of the time. I have never seen anybody with right side neglect, but I'm not saying you'll never see that. And you can break this into an output or an input. Output is I do not recognize visually, auditory, or whatever the left side of the world. It doesn't exist to me. I will not eat food on my left side of my plate. It doesn't exist. Okay, the left side of the world is gone to them.
If you turned their plate, so what was the right side is now the left side,
Yeah,
will they eat it?
They can eat that. There's all kind of techniques you'll learn. I'm not sure how much Sandra goes in for PT, but I'm assuming OT you do a lot of neglect kind of stuff. How to treat neglect, where to stand when you approach a patient who's got neglect. You of course want to stand on the side they don't recognise. You want to force them to recognise there is a left-hand side of the world. Okay. Where you put the TV in their room. All these kind of techniques you can use to try to get them into that left-hand side of the world. Mirrors and things. You can do some kind of interesting stuff with these folks. Output neglect is you do not use that output, don't output any motor commands to the left-hand side. You don't shave your left-hand side of the face, etc., etc. And this gets very severe in some cases, where they refuse to recognise they have a left side of their body, and it's a foreign object to them. And in some cases, people have actually tried to, cut their left hand off or cut their left arm off, amputate it, because it's some alien being attached to me. Okay, any questions about that? So that's just a brief overview of the words and a very general description of what these things mean. And in your later classes, you will go over the assessment and treatment of all these issues. All right. End of that. We're now going to look at our specialized receptors and follow these pathways into the cord and up.
When you talk about neglect, it said output or input. What does input mean? How does
Sensory, essentially sensory neglect. I don't hear, see, feel left-hand side of the world. It's basically, input, sensory neglect. I can't sense anything on the left-hand side of the world. Motor or output or motor neglect. I do not interact motorically with that other side of the world. I don't move my left hand. I don't into that space. Okay. These are the receptors we're going to go over. Come back to these. We'll go fairly quickly to the first few, just for completeness sake, name all these receptors within the skin, and we'll spend most of the time talking about the GTO, the Golgi tendon organ, and the muscle spindle. So the first thing we're going to look at, so here's a cross-section through the skin. We find all these first Merkel's discs, free nerve endings, myastus corpus, posterior corpus, emboli of Krauss, and the venous corpus. All those are basically within the skin somewhere. Okay? The GTO, of course, is in the interface between the muscle and the tendon itself. Okay? So the Golgi tendon organ lies in series with muscle fibers. The muscle spindle lies within the muscle itself. It lies alongside the muscle, lies parallel to the muscle itself. But we're going to go through these first few. Okay, so the first thing we're going to look at is these three nerve endings. These are basically the most undeveloped, unadapted, no structure involved with these three nerve endings. They're just a nerve ending, basically. The axon terminal ends in the skin somewhere. So these can give rise to various types of receptor. Perhaps we should mention that first. Broadly, there are two major receptors we will deal with as therapists, or are useful for us as therapists. Mechanoreceptors and thermoreceptors. There are also chemoreceptors, but we don't really, that's not useful to us perhaps. Chemoreceptors sense damaged skin, nerve damage, not nerve damage, but tissue damage. So those release chemicals and that damages, and these chemoreceptors will pick that up. Okay? But by and large, the ones that are most important to us as therapists will be mechanoreceptors and thermoreceptors. And particularly mechanoreceptors, I suppose, are most important to us. Mechanoreceptors, of course, respond to mechanical deformation. When you push on the skin, you touch the skin, pressure, stretch, vibration. These mechanical deformation of the receptors causes them to fire. Okay, mechanoreceptors. A subcategory of our mechanoreceptors are called proprioceptors. You've heard this terminology before, yes? I'm not going too quick on it, so you must have heard this terminology. Proprioceptors, what do proprioceptors do? They're a subset of our mechanoreceptors. What do they do? What's proprioception?
where our body is in space?
Where your body's in space, how your body's moving. So they're going to give you information, displacement of the muscles and joints. When I know where my arms are in space, when my eyes closed, that will be a proprioception. What's the difference between proprioception and kinesthesis?
Isn't kinesthesis how your body moves in that space versus knowing where it is?
Kin means what? Kinesiology, the science of movement, right. So kinesthesia are basically detection of your movement of the body. So how your body is moving in terms of the speed of the joint, how fast my elbow is bending, how fast my knee is bending, or whatever. Gives you a kinesthetic awareness of the motion of the body. Proprioception is kind of, they're often confused, used interchangeably, but essentially proprioception can give you static information as well. But kinesthetic essentially is a subcategory of proprioception in many texts, but sometimes they're used interchangeably. So this is very important to us if patients have lost proprioception, right? If lost proprioception, they're going to be impaired, depending on where they've lost that proprioception from. Our thermoreceptors, of course, are sensitive to heat and cold. So those are the ones we use, important to us in our modalities. Our ice packs, our hot packs, our whatever, ice massage will do for whatever. So those have been picked by thermoreceptors. We also have nociceptors, right? You've heard of that term? They are what?
Pain.
Pain. The nociceptors are basically a subset of our mechano and thermoreceptors that sense pain. A subset of mechanoreceptors and thermoreceptors. We also do, though, have something what's known as polymodal receptors. Polymodal nocioceptors. So nocioceptors usually are within your sense pain in terms of temperature, pain in terms of pressure, etc., etc. But some of these receptors actually respond to more than one modality. They're polymodal nociceptors. So they will signal pain through temperature and pressure and whatever. They're responsible for more than one mode of pain. Okay? And these polymodal nocioceptors give rise to C fibers by and large. And the pain, the sensation of pain coming out of our polymodal nocioceptors via the C fibers is this slow, dull, aching pain. You've heard have you gone over the differences between types of pain? Yes, no, yes? So you have this, they broadly separate pain into two categories. Whether this slow, burning, aching, dull pain and a sharp, prickly pain. Okay? The slow, throbbing, dull, aching pain comes via C fibers, via these polymodal nocioceptors. Okay? And that's going to be a C fiber, which is also known as what fiber in Roman numerals. What's a C fiber? Remember that question? That table in the thing tells you need to know the difference between the Roman numerals and the Greek letters. What are C fibers?
They're 3, right?
Type 4. They're the last one, aren't they? Type 4. They are the unmyelinated, slow conducting, small fibers. And that's the slow burning pain sensation. Our thermo and mechanoreceptors that give rise to type 3 fibers, okay, not type 4, type 3, give you your sharp, prickly pain. So C fibers coming out of various receptors give rise to the dull, aching, throbbing pain. If you transmit along a type 3 fiber, which is a faster conducting fiber, remember, a thicker fiber, type 3 fibers are known as what in Greek letters? A delta. The A delta fibers give that sharp, prickly pain sensation. So A delta 3, C also type 4. Non-myelinated they are.
I have a quick question.
Yeah.
What number is B?
B. What's type 2 B5? Which one are you looking at now?
Well,
A beta?
I'm looking off of the grid in the book, how it separates it. There's the 4 A's and then B and then 2 C's.
B fiber, what's B fiber?
That's the preganglionic.
Yeah, they're a class of its own. They are not on this category. They are the preganglionic neurons. Let me explain it to you later, perhaps there's a confusion there in that book. Which book are you looking at?
Just the neuron.
The table.
Yeah.
Yeah, let me come back to that and I'll explain that in detail. But these fibres are the ones arising from our receptors, and that depending on the type of fibre arising from them will determine what kind of sensation they convey. And the thickness of the fibre will give you the speed of the fibre. Okay? We won't explain that well. I'll show me what you're talking about, and then I'll clear it up for you, hopefully. Do you know how to distinguish between pain sensation? How do you do that? How do you distinguish whether the pain sensation is intact or not? So if somebody's lost pain sensation, how do you test for pain in a clinic? Do you stab them with a knife? Stick a pin in them until it hurts? No, what do you do? Sharp dull test, right? You familiar with that, OT, sharp dull? No? Basically, if you want to know that pain is working okay, that pain fibres, receptors, whatever, you will stick something like, I don't know, a paper clip into them, but not into them. Or a pointy pencil. Now you should, if pain fibres are going to work, a pain pathway is working, you can feel a sensation of sharpness. It doesn't have to be pain, just sharpness. If you don't have that sharpness feeling, you feel dullness, then you haven't got pain sensation working. The best example of this is, I don't know, you go to the dentist, right? You have your tooth numbed up, you have a cavity filled or whatever, and your lip goes all numb. You can get a paper clip or a pin and stick it in your lip, couldn't you? And you wouldn't feel pain. But could you feel it? Yes, you'd feel pressure. You'd feel a dull pressure in your lip. But you could stick a pin right in there and it won't hurt. I cannot detect sharp pin prick pressure. I can feel something pressing my lip, but I cannot determine that it's sharp. It feels like a dull, the eraser end of a pencil. And that's basically because you've got an anesthetic or an analgesic in your system that's cutting down pain. Okay? Alright, let's go back to these receptors. So we have these categories of receptors. Those category receptors, these receptors are going to be one of those categories essentially. Anareceptor, thermoreceptor, chemoreceptor. And the first, most undifferentiated receptor is this fella, the free nerve endings. So basically the axon terminus terminates in the skin. There's no receptor itself, it's just free nerve endings. And unfortunately, in different texts, you will see different sensations attributed to all of these receptors. Unfortunately, this is a mess. These receptors, you'll see a general agreement in most texts, but in some texts they'll say that does heat and that one doesn't, and it's a bit of a mess. So what I'm presenting here is what I think is the consensus. But you will find differences in some texts, I'm afraid. Your free nerve endings give rise to A delta, C, and A beta, which is a type 2 fibre. Okay? And these can give rise to many sensations. Pain comes out of A delta, which is C fibres. Cold and heat, temperature receptors. These are temperature receptors in some cases. These free nerve endings can sense temperature. And that's going to be a C fibre usually coming out of those. And touch. is also responsible for some sensation of touch, and that will be a C fibers, usually in some A delta. So you see mixed and
What was the first one?
pain, temperature, pressure. Those are all kind of sensitive attributed to these free nerve endings. There is a free nerve ending, a sort of very slightly modified free nerve ending that goes around the hair. Your hairs in your back of your hand and your arm or whatever. It's known as the root hair plexus, which is this wrapping around of the free nerve endings around around the hair root itself. Okay, these are free nerve endings that wrap itself right around the hair. These are obviously sense the pressure or the movement of the hair. And these are known as what's a rapidly adapting receptor. A rapidly adapting receptor. Do you know what I mean by a rapidly adapting versus a slowly adapting receptor? You've heard that terminology before? So a rapidly adapting receptor basically signals when it's touched, when it's prodded, when it's whatever, quickly. But if that touch, that pressure, that vibration, whatever it is, carries on, that receptor no longer signals. It signals initially and then turns off quickly. A slowly adapting receptor will tell you something's touching my skin for a long, long, long time. As long as it keeps touching you, it'll keep telling you. So, for example, get a pen or a pencil and find a hair on the back of your hand or on your arm and bend it over to 90 degrees. Bend it over, you can feel it. And now, a second later, you can't feel it. Right? If you touch that hair, you can feel it, the touch, instantly. But if you carry on with that hair bent over at 90 degrees, then you cannot no longer feel it, right? That's a rapidly adapting receptor. It said, I'm being touched, and that's it. It doesn't say I'm continuing being touched. It just turns off very quickly. A rapidly adapting receptor. So there's various types of these free nerve endings within the skin. They signal a whole variety of things. Merkel's discs, they are modified epidermal cells. They're on there somewhere. They're very minimally modified epidermal cells, and these give rise to a type 2 fiber, and they are a slowly adapting receptor that respond to pressure. So pushing in on the skin with your thumb or whatever, a pencil, pushing into the skin surface itself provokes Merkel's disc to give you the sensation of pressure. Quickly go through these because I want to get to the most important things at the bottom there, the GTOs, muscle spindles. Precinian corpuscles, these are very common. Precinian corpuscles. You can see there, type 2 fiber, a very rapidly adapting receptor. These are found deeper down, right deep down in the skin. You can see these bulbs down here, these big, they look like onions. They're lamellated, they've got lamina, they've got layers, they're deep within the dermis. So these layers of the onion, as it were, these connective tissue layers, are filled with a fluid between the layers. And the receptor is in the middle of all that mess. So it's an onion-like thing with little liquid between the lamina, which acts like a big damper. It dampens signals coming in. Okay? You find these very numerous in hairless skin. So in your fingertips, in your lips, in your eyelids, in the tongue. And these are very important in what's known as discriminative touch. What is it that's on my hand? Is it a drop of water? Is it a fly? Is it whatever? these Pacinian corpuscles. We're talking about Pacinian corpuscles. Can I skip one?
Yeah.
Where am I going backwards? Did I talk about Meissner's corpuscles?
No.
Okay, we'll come back to that. Meissner's corpuscles, Pacinian corpuscles. Let's finish this one. These are onions deep within the skin. And they are very responsive to vibration. Very rapidly adapting receptor. And it's those layers of the onion and the water, the liquid between them, act as a damper. It's like, let's say, okay, get a balloon full of water and you hold it on your hand. That's your Pacinian corpuscle. If I tap the surface of the balloon rapidly or push onto it, I feel that vibration going into my hand. I'll feel it. But if I press on that, I tap the balloon and hold the balloon down, I do not sense pressure on my hand, do I? Do you know what I'm saying? If you push on a balloon, you can feel that the vibration going through the balloon, but if I push on it and hold it, your hand underneath the balloon will probably not feel anything. Does that make sense? That kind of sensation of, because of the liquid and the layers within the Pacinian corpuscle dampen stuff down, as soon as the initial push is ended, you don't feel it. So these are very important in sensing vibration. They're very rapidly adapting receptor and they sense vibration. These are the Pacinian corpuscles. I'll get you confused there. Because what I meant to say about, okay, let's come back to the Meissner's corpuscles. These are the ones that are found in your hairless skin. So scratch that sentence I said about hairless skin being Pacinian corpuscles. Sorry about that. I was just skipping it around there. Meissner's corpuscles are the ones that are found in hairless skin. And these are the ones that are responsible for discriminative touch. I screwed that up there, didn't I? I kind of skipped ahead there. Okay, let's start again. Pacinian corpuscles, vibration, very rapidly adapting receptor. Okay, that's the main thing to realise. They're deep within the skin, deep down. Okay, those are Pacinian corpuscles. Those are the onion type things. The Meissner's corpuscles, these are more superficial in the skin, and these are a slowly adapting receptor, and they're the discriminative touch fellas. They'll tell you, for example, if you get a paper clip and you open it up, is there two things pressing against my skin, or is there one thing pressing against my skin? How wide do they have to be before you can see there's two things? So it'll tell you, discriminate between 1 and 2 points pressing on my finger, on my lips. Okay, so that gives you discriminative touch. Okay? Let's finish off these and get to our interesting ones. End balls of Krauss, cold receptors, temperature receptors generally speaking, usually cold, you'll see, type 3. I don't think we'll come across these much in therapy perhaps. Ruffinian's corpuscles, these are a slowly adapting receptor and they are thought to be, you'll see in text two different things ascribed to these, skin stretch and some say temperature. I mean, I've never done any research on this so I don't know what it is, but I just, generally speaking, you'll see Ruffinian's corpuscles being skin stretching sensation and maybe, and some text will say temperature associated with that. I confuse you there. We'll go over the next class again. But this is what we want to go into. This is dermatomes. We know dermatomes, don't we? You all know your dermatomes, don't you? Refresh your dermatomes. Yes. This is the GTO. This is more interesting to us as therapists. The Golgi tendon organ is found in the interface between the muscle, as the muscle, the collagen fibers of a tendon form or blend into the muscle itself. So the junction between the tendon and the muscle. And At either ends, of course. In both tendons, you'll find these Golgi tendon organs. Okay? They're named after, what's his face? Camillo Golgi. There's lots of things named after him. Like Golgi, you see Golgi all over the place in the body, don't you? Golgi, this, Golgi, that. He got Nobel Prize in 1906 for his work on neurophysiology. What happens here, okay? You have the tendon itself, the collagen fibers of the tendon come out of the muscle into the tendon, and they're all wrinkled. They're twisted. There's strands in it, like strands of spaghetti, kind of loose, limp spaghetti. Okay? That's collagen fibers. And within those collagen fibers, you find these nerve endings, these dendrites, twisting between between the gaps of the spaghetti strands. You can sort of see it, on that bottom right hand there. You can see the axon going between the spaghetti, the collagen folds, as it were. Okay? So, what happens then when you increase the force on the tendon? Those loose, floppy strands of spaghetti, of collagen, straighten. So they're crinkled at beginning with, to a certain extent, and as you stretch them, you put force onto the tendon, those collagen fibers straighten, and therefore they'll squeeze the axon between them, between the collagen fibers. So as they straighten, the gaps, the folds, the curves in the collagen straighten out and obviously compress the axon, the terminals that have threaded themselves between these wrinkles of the collagen fibers. And that, of course, signals force. So the more you stretch the straighter you make that spaghetti, the more force is applied to the axon, the more force the muscle the GTO is triggered. And the GTO is connected to a 1B afferent. The 1B fiber comes out of the muscle, out of the Golgi tendon organ, and goes into the cord. Okay? Which side of the cord does it go in? Back or front? Dorsal. Dorsal root. The cell body is found where? Where is the what type of neuron is this? Unipolar, bipolar, multipolar? Multi-bi-uni. Unipolar. They're all unipolar, remember, coming in. All the sensory neurons are unipolar, except the special senses, some of those are bipolar. So where do you find that nerve, the cell body? Dorsal ganglion. That is where you find those unipolar cell bodies. All those receptors we're talking about, every single one of those receptors, all those fibers we're talking about, all are going to be unipolar neurons with a cell body and a dorsal ganglion coming in the back side of the cord. So if you want to cut sensation from somebody, you cut the dorsal roots, they have no sensation, they are deafferented. Okay? So this fiber is going to come in, and where is it going to go? In the cord. It comes in the dorsal root, fine. Then what? How does it ascend to consciousness? This supposedly can sense 0.1 of a gram of force. So when I pick up a weight, oh that's heavy, that's light, whatever, I'm using my GTOs. And they're telling me how much force I'm making against this table, how much force I'm generating against this wall in my triceps. It's the GTOs telling me that. When you feel that, is this milk jug full or empty, it's the GTOs telling you, the brain, consciousness. How does it get to consciousness? What pathway? You've had this in the exam, right? Most of you got it right. Well, so. What is this sensing? This is proprioception, right? Proprioception. GTOs are one of our proprioceptors. How our body's moving, whatever. And so how does it get to my brain? Get to consciousness how? It's coming out of my arm. I'm holding a milk jug. I want to know how much milk's in this jug. I know, I can feel it's heavy. How is that done? GTO, 1B fiber, dorsal root, then what? fasciculus cunatus, nucleus cunatus, crosses over, media meniscus, second order, up to where? Thalamus, somatosensory cortex. We've done that 50 times, right? Proprioception of the dorsal columns, the posterior columns, right? That, because I've told you it comes from my arm, it's got to go up the fasciculus cunatus. If it came from my thigh, I'm feeling how much weight of a leg extension exercise I'm doing, how much force I'm generating. Where's that going to go? Fasciculus gracilis, nucleus gracilis, crossover, thalamus, to somatosensory cortex. If it's coming from my arm, whereabouts on the somatosensory cortex is it going to go? That strip over my head. Close to the center or on the side? How is your somatosensory cortex somatotopically organized? Where is the arm?
On the side.
On the side, here. Not in the crack. Therefore, it's the middle cerebral artery. Stroke would affect this, whatever, from my hand, for example. It's all linked together. You have to keep every bit of information together. You can't just compartmentalize blah, 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 blah. So, force sensation, GTOs. And we'll finish off by saying one thing this does. It's responsible for a little reflex, the GTO is. So here's my muscle, here's my GTO, here's the dorsal root, what does that mean? At high forces, that's what happens. What is that?
It's an inner neuron in the spinal cord, and it tells the muscle to stop.
It turns off the muscle. At high forces, the 1B neuron comes in, hits an inhibitory interneuron, which turns off the alpha motor neuron going to the same muscle that the 1B fiber just came from. So in other words, at high forces, the GTO will turn the muscle off. A protective response, perhaps. This is known as autogenic inhibition. Autogenic inhibition. The GTO wants to turn the muscle off that it just came from. So this is like in the Olympics, some bloke is going to go for the world record clean and jerk, and they slap him across the face, sniffing salt, come on, go for it, psyched up like crazy, comes up, tries to do it, and then his muscles collapse and he drops the bar instantly. The whole system gives way because you're about to snap your bones because I'm generating too much force, I want to break something, and my muscles just shut down. They drop the bar. It's too hard, heavy to hold, too much force, and it's your GTOs that do that to you. High forces, the GTOs, and it's debate how much high force there has to be. Some say it doesn't have to be too much force. There's always this kind of inhibitory influence of the GTOs on the muscle. What technique do you know from therapeutic skills uses that mechanism? PT should know this. When do you stretch a muscle and using this thingamajig? You hold, relax, contract, relax, PNF patterns, right? So what you do there, basically for OTs, so let's say you're contracting, stretching the hamstrings, right? You have the person on the back, you bring their leg up the hamstring and it's hurt, you hold it there. And what you have the patient do then? They contract against you. They try to contract as hard as they can in some situations the hamstrings to push the leg back down. You stop them. You stop them moving. The leg's contracting against the arm, against your hand or your shoulder, whatever you're doing, and then you tell your patient to relax and you crank the leg much further, can't you? Have you tried that? Have you done that yet? It's worked. So what's that you're doing? You're generating this force in the muscle. You're stimulating your GTOs. The GTOs, what do? They turn off your hamstrings, which are the muscles you're trying to stretch. So it allows you to stretch them more. Proprioceptive neuromuscular facilitation techniques. So the GTOs are very important in many of our hold relax, our PNF patterns, whatever. I don't expect the OTs to know that, but the PTs should know what this is, the value of having one of these thingamajigs in your muscles. Okay? Alright, let's stop, because the muscle spindle is more complex. The muscle spindle is 10 times more complex than this, unfortunately, because it has both motor and sensory components to it. Right, exam. Jason, do you shout your names out? Chris, would you mind shouting your names out?
Sarah Pelletier. She's over there.
Brittany Rex. Where's Brittany?
Right here.
Gwen Maddox.
Don't all know your names yet. Ashley, where's Ashley? Ben. You leave it?
What was that?
What was that? What was that? Nick. Yep. Jen, Rachel.
Hey,
Sarah Bolton. Rena.
She's over here. Chantel's here. Jason. I have somebody else.
I guess your name.
Okay, pass it down.
Let's see.
Jamie Wallace.
Sorry. I don't know his name. She goes back. I'm terrible with last names. Come on now.
Misty.
Caitlin Lambert.
Jason Phelps. Where?
Ryan behind you.
Ken.